Right now, super speedy, we're gonna go over the whole entire process of glycolysis. And basically what glycolysis is, this is how you get energy, ATP, out of glucose, out of sugar. And this is done in your muscle, in your brain, in your red blood cells. This can be done pretty much in any cell of your body, and this is how you get energy from sugar. First, obviously, this is glycolysis, sugar burning. So you start with glucose. You start with glucose at the top, and then what you have to first do, you have to get the glucose into your cell. Let's just say this is a muscle cell and you're like working out. Firstly, insulin is going to knock on the cell door and say like, hey, wake up, we have a ton of sugar in the blood. You need to get that sugar into your cell so you can use that sugar to make energy. So high blood sugar, that stimulates insulin. Insulin at the cell is gonna stimulate the transport of a GLUT4 glucose 4 transporter to the cell membrane. So when insulin like taps on the cell, says, hey, we're here, you put this GLUT4 transporter, this doorway for glucose in the cell membrane, and now glucose will enter the cell. So let's just sequence this out. You eat sugar, blood sugar spikes, that causes insulin to spike. Insulin tells your muscle cell, move the door, like open the door for glucose to come in. That's the GLUT4 transporter. Now glucose comes into your cell. First thing we do when we have glucose in the cell is we phosphorylate it. When we are phosphorylating glucose in a muscle cell, we are using hexokinase. Hexose because it's a 6 carbon sugar. So hexokinase because it is a kinase. Kinases phosphorylate things. So we go from glucose plus hexokinase uses ATP to phosphorylate glucose, and now we have G6P, glucose 6 phosphate. After we have, I'm looking at my notes behind me. After we have glucose 6 phosphate, we then use phosphohexoisomerase. That's an enzyme. Phosphohexoisomerase, F6P. And you can guess what phosphohexoisomerase does. It isomerizes phosphohexoses. So now we've gone glucose G6P to F6P. F6P is fructose 6-phosphate. Once we have fructose 6-phosphate, we have to rearrange that into the next molecule in glycolysis, which is F16BP, fructose 1,6-bisphosphate. So this is another phosphorylation enzyme, so we're using another molecule of ATP. It's an investment. And we use the enzyme PFK1, phosphofructokinase 1. So it's kinase, it's phosphorylating phosphofructose. So fructose with a phosphate, F6P, gets phosphorylated, and now we have F16BP. Once we have F16BP, after here, everything doubles because what we actually do at this step, we have F16BP. Recall that we started with the 6 carbon molecule, and now we have a phosphate on the first carbon and on the 6th carbon. So F16-bisphosphate means on the first carbon, number 1, the F16P, there's a phosphate. On the 6th carbon, there's a phosphate. What we do now after we have F16BP, is we chop it in half. Here we use the enzyme enolase, and this gives us GA3P, glyceraldehyde 3-phosphate, and DHAP, dihydroxyacetone phosphate. So we go F16P, the enzyme enolase chops it in half, and now we have glyceraldehyde 3-phosphate plus DHAP, dihydroxyacetone phosphate. we have a teeny weeny little problem because we cannot send DHEP down the rest of glycolysis, but we can send GA3P down the rest of glycolysis. So the next enzyme after this step is glyceraldehyde 3-phosphate dehydrogenase, GA3P dehydrogenase, GA3PDH. GA3P dehydrogenase will only act on GA3P. What we need to do is we need to rearrange DHAP into GA3P. So here we use the enzyme triose phosphate isomerase, triose because it's a 3-carbon sugar, phosphate isomerase because it's got a phosphate attached. So we use the enzyme triose phosphate isomerase to isomerize DHAP into GA3P. GA3P. And this is, where the frick is my colored marker? I have no idea. This part is super important because now we have 2 of everything. Remember, we had one F16BP, we cut it in half to yield GA3P and DHAP, and then we turn the DHAP into GA3P, and now we have 2 GA3Ps. So everything after this step is times 2. Once we have GA3P, we use GA3P dehydrogenase, and guess what that yields? It's a dehydrogenase, so it dehydrogenates GA3P, it oxidizes GA3P, and what does it reduce? NAD+. NAD+ is reduced, yielding NADH, and NADH can float off into the mitochondria and function in the electron transport chain and give us some ATP. So let us draw that out right this very second. GA3P, the enzyme GA3P dehydrogenase, GA3P, glyceraldehyde 3-phosphate, is oxidized, dehydrogenated, and NAD is reduced into NADH. Is any energy yielded in this reaction? Nope, but we do add a phosphate. Okay. In this enzymatic step, a phosphate is also added. So I'll show you what we have going on here. GA3P plus NAD. So let me show you what we have going on here. Glyceraldehyde 3-phosphate, NAD+, and inorganic phosphate, PO4 3-. Glyceraldehyde 3-phosphate dehydrogenase oxidizes this molecule and reduces this molecule. This step yields NADH and 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate, and it is 1,3-bisphosphoglycerate because GA3P has 1 phosphate and we've just added another phosphate. So now there's a phosphate on the first carbon and on the third carbon, and we've just yielded some NADH. Because everything below here is happening twice, we've yielded 2 NADH in this step. After we have 1,3-bisphosphoglycerate, the next enzyme we use is phosphoglycerate kinase. What do you think happens here? Phosphoglycerate kinase. Because it is a kinase, something is getting phosphorylated. What actually happens is we remove a phosphate from bisphosphoglycerate. So instead of having 1,3-BPG, now we have 3-BPG. So this is the first energy yielding step in glycolysis. We've just made 2 molecules of ATP when we go from 1,3-BPG into 3-PG. And this is done by the enzyme phosphoglycerate kinase. We take a phosphate off here, we phosphorylate ATP, and this gives us the molecule 3-PG, 3-phosphoglycerate. Everything below here happens twice, so we've yielded 2 ATP molecules. We're so close to the end, we're almost done. Now we have 3-phosphoglycerate. The next enzyme we use is, whatever enzyme we use, we have to get the molecule PEP, phosphoenolpyruvate. I have no idea what enzyme that is. I'm looking at my notes. Oh, okay, no, I lied. After 3-PG, 3-phosphoglycerate, we use phosphoglycerate mutase. It's a mutase that literally just means it's rearranging the chemical organization. So we go from 3-phosphoglycerate to 2-phosphoglycerate. That's this step right here. Now we have 2-phosphoglycerate. We're super, super close. Next thing we need to do is we need to make PEP, phosphoenolpyruvate. So we use the enzyme enolase. We remove the equivalent of a water molecule, and we go from 2-phosphoglycerate to phosphoenolpyruvate, and we remove a water molecule. Now that we are at PEP, this is the last step in glycolysis. We use the enzyme pyruvate kinase to yield pyruvate. This is an energy yielding step, and we make ATP. So when we go from phosphoenolpyruvate, we use the enzyme pyruvate kinase. We dephosphorylate PEP, and we yield pyruvate. So ADP goes in, ATP comes out. Phosphoenolpyruvate comes in, pyruvate comes out. This is glycolysis. What happens next is if we are doing aerobic oxidation, so making energy in the presence of oxygen, this pyruvate will travel into the mitochondria. It will encounter the pyruvate dehydrogenase complex. It will be oxidized. And then we want to yield acetyl-CoA. Acetyl-CoA enters the tricarboxylic acid cycle, and then that pops out NADH, FADH. The NADH and the FADH travel into the mitochondrial matrix, and they drop electrons off along the electron transport chain. These electrons bump from complex to complex to complex to complex. They create a proton gradient, and this proton gradient is coupled to ATP synthesis. ATP synthase, the enzyme, phosphorylates ATP synthase, the enzyme, phosphorylates ADP into ATP so long as we have a strong proton gradient. And that proton gradient is generated by NADH and FADH dropping their electrons and protons off at the electron transport chain. Back to glycolysis. This is the complete process of glycolysis. If there is no oxygen present, your cells will be relying entirely upon this pathway to make energy, and then pyruvate will be fermented into lactic acid. And then that lactic acid can enter your blood, travel back to your liver, and your liver can use that lactic acid to do gluconeogenesis, the synthesis of new glucose. Right now, let's just finish glycolysis. What have we done? We invested 1 glucose, 2 ATP molecules, and 2 NADH molecules. So let's say in 1 glucose, 2 ATP, 2 NAD plus. Into glycolysis, 1 glucose, 2 ATP, 2 NAD plus. What did we get out of glycolysis? Out. Everything down here after this step. Oh, this enzyme is actually called aldolase. I messed that up. I'm sorry. Okay. Everything below aldolase is doubled. 2 NADH we got. 2 ATP here. 2 ATP here. So the outputs of glycolysis are 2 NADH, 2 ATP at phosphoglycerate kinase, and 2 ATP at pyruvate kinase. So we got a total of 4 ATP, and then we also got 1 molecule of pyruvate. So the inputs here, the outputs here. The net ATP yield of glycolysis is 2. We invested 2 ATP because we had to phosphorylate glucose, and then we had to phosphorylate fructose 6-phosphate into fructose 1,6-bisphosphate. And then down here, we yielded 4 ATPs. So we got 4, we invested 2, 4 minus 2 equals 2. The net ATP yield of glycolysis is 2 ATP, but we also got 2 NADH as well. If oxygen is present, then that NADH will travel into the mitochondria and contribute to ATP synthesis at the electron transport chain. And that pyruvate will also enter the tricarboxylic acid cycle. It will produce NADH and FADH, which will also enter the electron transport chain. And the net yield of aerobic respiration, oxidation involving the mitochondria, is like 32 ATP. The net yield of glycolysis is 2 ATP. That is like so bad, like really not good. Your body would way rather oxidize glucose and fatty acids to get 32 ATP than it would to get 2 ATP from glycolysis. So glycolysis is kind of like desperado situations. But this is the entire process of glycolysis. And I really hope you enjoyed this study session. If you did, please like, comment, and subscribe. It helps me put more time, energy, and resources into making these videos and into making petite little freebies for you guys. So I hope you enjoyed, and I'm gonna post this in 5 minutes. And have such a lovely day. Goodbye. Mwah.